Good day, everybody, and welcome to this webinar. This is the third of a series of webinars titled Microbiota and Gut-Brain Connection, a New Frontier in Neurogastroenterology. And these webinars are organized by the European Society of Neurogastroenterology and Motility together with Bromatech. And it's a great pleasure and an honor for me to be with you tonight for several reasons. First of all, because we have the pleasure and the honor of having two stars, I would say two extraordinary scientists tonight, Professor Stephen Collins and Professor Alessio Fasano. And I would say that they are among the brightest stars in the galaxy of gastroenterology. I've known them for quite some time, and they are not only great scientists, but also good friends. And in particular, Professor Stephen Collins has been also my mentor sometime back in the 90s when I was joining his group at McMaster University in Hamilton, Ontario. And also another reason why I'm so happy tonight is that we are three of us Italians. Maybe Steve is half Italian because his mother was from Italy originally. And Alessio Fasano is definitely Italian, even both of them work, Collins in Canada and Alessio in the United States. So, the housekeeping news that I have to give you is that what you can do is during the seminar is to place your questions to the speakers in the chat of the Zoom platform whenever you want, and then we will select these questions for the question and answer at the end of the presentations, at the end of the seminar. Also, another housekeeping information is that you can have simultaneous translations, and here you can see some information that you want to go through in order to get the right translation into your language, particularly in Spanish and in Italian. To follow this webinar in English, do not change your Zoom audio settings. To follow the Italian or Spanish simultaneous translation, click on the interpretation icon and then choose your language. You can then choose mute original audio to mute the background English audio. And on mobile phones and tablets, you may need to tap more to access the interpretation icon. It is advisable to install the Zoom app instead of using the browser. So thanks again for joining us tonight. And I would like to introduce the first speaker of the webinar tonight, Professor Stephen Collins. He is the director of the Farncombe Family Digestive Health Research Institute at McMaster University in Canada. And as I said, he is a good friend, but a great scientist. And he's a pioneer, I would say, in the field of gastroenterology. And his research is focused mainly on the connection between the gut and the brain, and particularly the connection between the microbiota and the brain. And he has started these experiments in animals, laboratory animals, but also his research is focused on digestive conditions such as irritable bowel syndrome. So we are very much looking forward to hearing to his presentation tonight that is titled Microbiota-Brain Axis in Irritable Bowel Syndrome. Thank you very much, Steve, for joining us, and please go ahead with your presentation.
Okay. So Giovanni, thank you very much for that kind introduction, and also thanks to the organizers for this invitation to present to you on the topic of the microbiota-brain axis in the context primarily of the irritable bowel syndrome. During this talk, I'll cover three topics. First of all, I'll discuss the experimental evidence supporting the existence of this axis, which is a bidirectional axis. Then I'll try to put this in the context of its relevance to irritable bowel syndrome. And in the last part of the talk, I'll talk about the therapeutic implications of manipulating this axis in the treatment of IBS. As most of you know, the irritable bowel syndrome is the most common of conditions, GI conditions on a global basis. It is characterized by abdominal pain and altered bowel habit. It's well known to be heterogeneous in terms of its clinical presentation, but that heterogeneity also applies to the underlying pathophysiology. And even within clinically homogeneous subgroups of IBS, one will see heterogeneity in terms of the underlying pathophysiology. The irritable bowel syndrome, as some of you well know, does involve the gut-brain axis, and the involvement of the brain is really based on is based on the knowledge that between 50 and 90% of irritable bowel patients seeking treatment have comorbid psychiatric disorders. And these include mood disorders like depression and anxiety, as well as somatization. In addition, psychological distress or stress is an independent risk factor, not only for the development of IBS, but for the exacerbation of pre-existing irritable bowel symptoms. So these are the reasons then why we invoke the gut-brain axis in the pathogenesis, pathophysiology of IBS. So what is the rationale, therefore, for implicating the microbiota in IBS? And it's the following lines of reasoning that I offer at this point in the talk. First of all, factors that we know precipitate or exacerbate IBS are also known to influence the microbiota composition and or metabolism. These factors include antibiotic exposure, particularly repeated or extensive antibiotic exposure, enteric infection, dietary change, and psychological stress. The second line of evidence is that changes in the composition of the microbiota or its metabolic activity, particularly in short-chain fatty acid production, have been demonstrated in subsets of IBS patients. And thirdly, as I'll point out later in the talk, an IBS phenotype can be induced in germ-free mice simply by transferring the microbiota from an IBS patient. Like the gut-brain axis, the microbiota-brain axis is bidirectional. So gut microbes can influence the brain in terms of its development early in life, its function, and therefore behavior later in life. And we also know that stress can alter the microbial composition of the gut, and I'll be going into both of these during this talk. So what is the evidence to support this gut-brain, this microbiota-brain axis? This is an axis, as you can see at the bottom, that's drawn a lot of attention from the public. This was on the front of Forbes magazine a few years back. Everybody's interested in how and why microbes can influence the way we think and the way we behave. But when we look back and remember hepatic encephalopathy, where clearly gut microbes, albeit in a pathological situation, can influence brain function, resulting in cognitive dysfunction all the way through to deep coma, and that this can be corrected by administering antibiotics to the gut. So this is really the first, I think, and the most long-standing and robust clinical evidence or observation that supports the existence of this axis. Most of the experimental evidence I'm going to show you occurs in preclinical work in mice. And the reason for that is it's much easier to manipulate the microbiota in an animal model than it is in man, where it's almost impossible. And we use this, we use a facility, a germ-free facility, sterile facility, where we can strictly control the microbial environment to prevent contamination. So at any point in time, we know exactly what the microbial status is of any animal under study. This, of course, limits what we can do in terms of the sophistication of the testing for behavior, because the apparatus has to be able to fit into the facility, number one, and number two, we have to be able to sterilize it on a frequent basis. The principle underlying behavioral testing in mice really relies on a balance between its inherent caution or its natural curiosity. It's a combination of both, just as in man. And when the mouse exhibits a lot of caution, we call this anxiety-like behavior. On the other hand, when it really doesn't show much caution, it lets its curiosity get the better of itself, we call this risk-taking behavior. And you'll see examples of this as we move on in the talk. So there are 4 tests that we're going to be using during the course of this talk that identify anxiety-like behavior and depression-like behavior. For anxiety-like behavior, we use 3 tests. One is a step-down test, where we put mice on an elevated platform, and then we measure the time it takes for them to go down, to jump down, and explore the environment. And the latency to step down is a measure of whether or not there is anxiety-like behavior. The light box, dark box test is shown here. Mice naturally prefer to be more comfortable in a dark environment, but their curiosity forces them to come out and explore a light environment. And the number of times that they come out here is what we measure, how often they come out and how long they spend. When the mouse is very anxious, it usually stays for a long time in the dark part of the apparatus. This elevated maze test is, as the name implies, is elevated above the floor. It has 4 limbs. 2 of these are covered, they're dark, and 2 of these are open. Naturally, the mice first gravitate to the dark parts of the limb, but every once in a while they'll take a chance and they'll risk coming out onto the open lanes. And a measure of anxiety is their avoidance of these open lanes. When it comes to depression, there are lots of tests, but as I said, we are constrained by the environment of our AGU facility. The test that we use is called a tail suspension test. Here the mice are suspended by their tail, and under normal circumstances, a mouse will spend very little time in correcting its posture so that it becomes upright, as you can see trying to do here. When a mouse is depressed, it adopts an attitude of helplessness, and it really makes very little effort to get back to its normal posture, normal position, and just remains immobile. And so the time it remains immobile is a measurement of depression-like behavior. So in terms of the evidence supporting this microbiota-brain axis, the first step was really to see, does the presence of a microbiota make a difference to behavior? And to do this, we study mice that are born without bacterial colonization. and we compare those to fully colonized mice, colonized, and we call these mice in this throughout this talk, we'll call them SPF because they're colonized with a normal microbiota that is free from any specific known pathogens. So in the absence, as I'll show you in a minute, in the absence of a microbiota, mice behave in a very abnormal way. They take risks. They seem to be disinhibited. Whereas mice with a stable microbiota behave much in the way that I've described before, a balance of caution and curiosity. So using this elevated maze apparatus, we studied germ-free mice and SPF, colonized mice. And as you can see down here, the germ-free mice spent more time in the open limb. They weren't afraid, and they spent a lot more time out here compared to normal mouse that really spent most of its time inside the closed area. So the germ-free mice was taking risks and spending more time on this dangerous open elevated limb. This change in behavior, this behavioral profile rather, of germ-free mice was accompanied by changes in brain chemistry. As you can see here, there was a reduction in the 5-HT1A receptor, which is a receptor for serotonin. This was reduced in the dentate gyrus region of the hippocampus, as you can see here. And another important mediator of brain function, which is brain-derived neurotrophic factor, was actually increased in that region of the hypothalamus. When these germ-free mice were colonized, these changes reverted to normal if the colonization was done within a few weeks of birth. There comes a time when this is not possible to reverse, but within the first few weeks of life, these changes are all reversed simply by colonizing the GI tract with microbiota. And what we found is it takes only one strain of bacteria to normalize this abnormal behavior. And I'll be coming back to this a little later in the talk. Let me give you an example of what this looks like. So this is an SPF mouse, a normal mouse, and it's put onto this elevated maze. And you can see that it spends a lot of time sniffing around, figuring out whether it's going to look. And this takes approximately 54 seconds to achieve. So rather than wait 54 seconds, I think you get the message. In contrast, the germ-free mouse will spend much less time because it's a risk taker, it's disinhibited, and very soon, after only 16 seconds, it will jump down and explore its surroundings. 16 seconds in the germ-free mouse compared to 54 seconds in the step-down mouse. So in very different behaviors, depending on the presence or absence of a microbiota. The second line of evidence addresses the question, can behavioral phenotype be transferred via the microbiota? And to address that question, we exploited differences between common laboratory strains of mice. So we know that different lab strains, different lab mice strains have different behavioral profiles. And we also know that even under the same conditions, environmental conditions, they have subtle differences in terms of their microbiota. This is the impact of the host on the microbial composition in the gut. So here we have the NIH Swiss mouse, which the students and fellows in the lab don't like because they are aggressive, they bite a lot. In contrast to the BOWEL mouse, which is a nice, pleasant mouse to work with, and it's very, very calm. Here you can see just the subtle differences in microbial composition between these two strains of mouse. So what we wanted to do here was to take this aggressive germ-free Swiss mouse and provide it with the microbiota from the bulb C mouse, which is calm, in the hope of calming down this NIH Swiss mouse and making it perhaps less aggressive. The results are shown here. The test again is this elevated platform, and we're measuring the latency to step down. This is the Swiss mouse. It takes only 20 seconds for this aggressive mouse to jump down and decide to explore its environment. If we take an NIH Swiss mouse that's germ-free and we colonize it with the microbiota from another NIH Swiss mouse, in other words, an autologous transplant, it makes no difference to its behavior. So the process of colonization itself does not influence behavior. However, when we colonize this mouse, this Swiss mouse, with the microbiota from the calm bulb C mouse, the behavior changes. And instead of spending just 20 seconds, it waits for almost 60 seconds, almost 3 times as long, before it decides to step down. So simply by transferring the microbiota between these species, we've been able, between these strains rather, we've been able to modify their behavior. And this modification of behavior, again, is accompanied by changes in brain chemistry. And an example here is, again, brain-derived neurotropic factor in the hippocampus, which is reduced as the mouse gets more calm. The third line of evidence addresses the question, can a subtle shift in the microbiota composition influence behavior in a healthy adult mouse? And this has more relevance to when we start to relate these lines of evidence to the clinic and to humans, because here, in contrast to the previous 2 examples, we're not dealing with germ-free models. We're taking a healthy adult mouse with a stable microbiota, and we're going to give it antibiotics to produce a small, subtle change in the composition of the microbiota, and to see whether or not this is accompanied by an alteration in behavior. So the mice receive a combination of 2 antibiotics and an antifungal agent to prevent overgrowth. And they receive this for 14 days by gavage. And at the end of the 14 days, we culture anaerobically and aerobically the bacteria. And there's no change in the total number of culturable bacteria. However, when we look at the composition, you'll see subtle differences in terms of the relative proportions of various strains of bacteria between the 2. So what we've done here then, we haven't decimated the microbiota, we've simply shifted the composition in a subtle way. This results in a change in behavior. Again, the step-down test here, this is the control before the antibiotics. With the antibiotics, there's a noticeable, significant change in the latency to step down. The mice become less cautious, they quickly step down. But when we stop the antibiotic and wait 2 weeks and the microbiota reverts to normal, this behavioral change is normalized. When we look at the light box, dark box test, the amount of time that the antibiotic-treated mice spend in the light box is increased compared to baseline, and again tends to revert towards normal when the antibiotics are withdrawn. The number of zone entries into the white zone is increased. So together, these findings said that when you've made a subtle change to the composition of the microbiota, it results in a measurable change in behavior. In this case, much more gregarious, curiosity-driven, risk-taking behavior after the administration of the antibiotic. And this is accompanied by changes in BDNF in the amygdala and in the hippocampus. So those are the 3 lines of evidence then that support the existence of a microbiota brain axis. What about underlying mechanisms? There are many mechanisms that have been described, putative mechanisms whereby molecules from various sources can influence brain function, many of them originating from interactions between the microbiota and the host or from the microbiota themselves. But the main ones of interest are tryptophan metabolites, vagus nerve transmission, microbial metabolites such as short-chain fatty acids, and innate immune activation. But one thing to remember right off the bat is that the involvement of these mechanisms really depends on the model that's being studied. And models that use, for example, probiotics have different mechanisms than models where you're manipulating the microbiota. We're particularly interested in the role of the immune system in microbiota to brain communication. And to do this, we have used various immunodeficient mice and used a monocolonization of these germ-free immunodeficient mice or wild type. And the reason we use monocolonization is because these immunodeficient mice often cannot stand a full colonization with a complex microbiota. They simply die. So we had to revert to a monocolonization, and in the studies that I'm going to show you, we used a wild type E. coli. Again, here we're using a light box, dark box. This is the germ-free mouse that spends a lot of time in the light box. Then following monocolonization, you'll see that it gradually, this behavior, this gregarious, curiosity-driven behavior is normalized. However, when we knock out the adapter proteins that are required for activation of innate immunity, we fail to see this normalization of behavior. So that the normalization of behavior in this model absolutely requires the integrity of the innate immune system. When we look at the adaptive immune system, and here we're using SCID mice, that is to say mice with severe combined immune deficiency that lack humoral and T-cell mediated immunity. Again, looking at the light box, dark box paradigm, again, here's the germ-free mouse showing a lot of curiosity. And over the course of time, as it's colonized, this normalization of behavior. When we did the same experiment in mice with no adaptive immune responses, we saw this normalization. So the normalization of behavior in the germ-free mouse is critically dependent on the integrity of the innate immune system, but does not require the integrity of the adaptive immune system. Currently, our working model is to further explore the mechanism between this communication. Clearly, there is innate immune activation involved down here. We believe that there is migration of dendritic cells involved. This is work in progress, and that up in the brain, that ultimately there is modification of the activation status of the microglia in the brain. So this is where our research is currently going. I do not have any data to show you in this regard, but this is the direction I think that we are taking. The gut-brain axis is bidirectional, and so is the microbiota-brain axis. And to illustrate this, I'll show you some data on the effect of psychological stress on the microbiota. So we are, sorry about that. So we apply psychological stress and see what the change is in the microbiota. Now, this was first done a long time ago, in the last century, in fact, where Mike Bailey and his colleagues at Ohio State were studying rhesus monkeys. And to induce stress, they would take infant rhesus monkeys away from their mothers for a few hours every day. And this would produce different degrees of stress in different infants. And they measured by culture in those days lactobacilli. And they found that the degree of stress correlated well with a reduction in lactobacilli number, the culturable number in the stool. So this was the first demonstration then that psychological stress could change the microbial composition. You could perhaps argue that there are other interpretations here, that the reduction in contact at an early stage in life could also account for this. But as I'll show you, there are other studies that have been done, again by Mike Bailey, showing that social stress, which is where a community of mice in a cage are introduced to a much larger mouse from another area, this produces hostility and aggression, and it's a form of social stress that's well described. This resulted in a change in microbial composition of the gut, and in that particular study, evidence of some immune activation with an increase in circulating IL-6. In our work, we use maternal separation in mice to show that the stress that this produced altered gut function, and it changed the microbial composition of the gut. And the behavior of the pups when they were maternally separated could actually be transferred in part simply by taking the microbiota and putting it into germ-free mice from these separated young mice. And we were able to show that part of the stress phenotype could be transferred via the microbiota. Difficult to do these studies in man. The first attempt was back in 2008, when they took the usual human laboratory animal, which is a medical student. Medical students were doing examinations over a period of 1 to 2 weeks. And during this time, their stools were collected, and they were found to have marked changes in the composition of the microbiota. And this was attributed to the examination stress. But we all know when you're doing exams for 2 weeks, your dietary habits and a few other things change, so that the interpretation of this is quite open. More recently, in pregnant women, a study was done, better controlled, to show that stress and anxiety in pregnancy results in a change in the microbiota of the mother. And of course, this has implications for the well-being of the offspring. In one of our studies, we studied the stress response, in which corticotrophin-releasing hormone is part of the hypothalamic pituitary activation. And we injected CRH into the brain, directly into the brain of mice. And we found that it altered colonic physiology, resulting in more pellet output. And this was accompanied by a shift in the microbiota composition, in this case measured by denaturing gel electrophoresis. So another example of how stress and stress-related chemicals might change the composition of the microbiota. So putting this together, the proposed mechanisms would be that you apply the stress. This induces activation of the hypothalamic pituitary axis, the release of CRH, which, as I've said, changes colonic physiology, which provides then a different physicochemical habitat, which in turn selects for different bacteria, and this produces a shift in the microbial profile. There's also sympathetic outflow, and we know that catecholamines have the ability to interact directly with the microbiota and interfere with communication between the microbiota to bring about or to contribute further to this shift in the microbial profile. Let's go back now to irritable bowel syndrome and the basis for implicating the microbiota in this common condition. So there have been a number of studies over the last 10 to 15 years, usually using 16S profiling of the microbiota, showing that there are changes in the microbiota in at least a subset of IBS patients. And the more consistent findings are reduced microbial diversity, instability of this profile over the course of time, and abnormal metabolism in some cases, particularly in terms of short-chain fatty acid production. So while these compositional changes occur, nobody has yet identified a microbial signature or a microbiotal signature that will confidently tell you this is IBS. We're not there yet. However, we can make some generalizations, and a recent systematic review from McMaster, from Paul Moietti and his group, reviewed the literature and made some generalizations that I think are helpful. They found that overall, Bifidobacterium and Faecalobacterium were reduced, and certain Lactobacillus species, Bacteroides and Enterobacteriaceae, were increased. These are generalizations. I don't know if systematic reviews can apply as easily to mouse work as it can, to this kind of work as it can to other forms of systematic review, simply because of differences in terms of the depth of sequencing and so forth. But nevertheless, this is a generalization, but I want to emphasize that there is no microbiota profile that I know of as yet that will confidently give you an indication that this is IBS. So the data that I've shown you so far really provides an association between IBS and the microbiota. So the question emerges, is there any causal relationship? Does the microbiota actually contribute to the IBS phenotype? To address that question, what we did was we took some IBS patients, took their microbiota and gavaged it into germ-free mice to see whether we could produce in that germ-free mouse an IBS-like phenotype to provide some sort of proof that the microbiota at least has the potential to contribute to the phenotype of the patient. So this is the protocol. We took 6 diarrhea-predominant IBS patients. 4 of those had comorbid anxiety with fairly high anxiety scores using this index here. And 2 of them had no anxiety, they simply had diarrhea. And we had 5 healthy controls that had neither GI symptoms nor psychiatric problems. The stool was collected anaerobically and then gavaged into from each donor into a minimum of 10 germ-free mice. We allowed the microbiota to colonize for 3 weeks. And this would eliminate any host factors that were present in the stool, and then measured a gut motility, secretion, permeability, behavior, and the immunological profile. This slide here shows the effect on motility. To assess motility, we basically measured transit. We used steel beads, small steel beads that were gavaged into the mouse. And 3 hours later, we took an X-ray to see where the beads had arrived. Most of these beads remained in the cecum in the mice that were colonized with control microbiota from a healthy subject. But when we colonized with the IBS microbiota, only 1 bead remained, indicating that the others have been expelled, indicating fast transit, and you can see that it's quantified here. So that microbiota from diarrhea-predominant IBS patients induces rapid transit in the recipient mice. We next looked at epithelial function and looked at paracellular permeability using chromium EDTA fluxes. And we found that in the colon, but not the jejunum, that there was an increase in permeability in the mice that had been colonized with the microbiota from the IBS patients. We also looked at the secretory profile of the epithelium, looking at iron secretion. And again, we saw no changes in the jejunum, but in the jejunum, there was a significant increase in iron fluxes, indicating a secretory profile of that epithelium, which is consistent with the phenotype of the diarrhea patient. So, microbiota from diarrhea-predominant IBS patients alters epithelial function in the colon, resulting in alterations in paracellular permeability and in terms of secretion. We next measured behavior. And again, we used the step-down test and the light box, dark box test. And what we found was that when the mice were colonized with IBS microbiota from patients who had no anxiety, there was no change in the performance of the step-down test. When we colonized mice with the microbiota from IBS patients with diarrhea who had high levels of anxiety, the profile of the step-down test was abnormal. The mice became more cautious, more anxious, because they took a longer time to step down from the pedestal. In the light preference, in the light box, dark box apparatus, we found a similar profile. The microbiota from IBS patients without anxiety produced no change in the performance here. But the microbiota from the patients who had high levels of anxiety spent very little time in the white box. They were anxious. They remained more time in the dark box. We measured the total amount of activity that these mice exhibited, because one concern was this was a general malaise effect, but this is not the case because the locomotor activity did not change across these three groups. So that the presence of anxiety in the donor patient could be transferred through the microbiota to the recipient mice. But no anxiety was transferred if the donor patient had no anxiety. This slide here shows the evidence that there is some degree of immune, apologies again, immune activation. So again, we looked at CD3 cells in the lamina propria of the colon. And when we transferred the microbiota from IBS patients without anxiety, we saw no increase. But when we transferred from the IBS patients with comorbid anxiety, we saw a significant increase in the number of CD3 cells. We measured beta defensin in the colon, a measurement of innate immunity. And again, the same profile. It was only when we colonized these mice with microbiota from patients with anxiety that we saw evidence of innate immune activation, as reflected by an increase in beta defensin 3 secretion. We used nanostring technology to look at gene expression in the colon of these patients. And as you can see, it was the IBS patients with high levels of anxiety. Their microbiota tended, this is a heat map of these genes here that are related to immune regulation, were upregulated, whereas in the IBS patients with no anxiety, there was very little evidence of this activation. So microbiota from IBS patients with anxiety induces immune activation. There was no increase in myeloperoxidase or in pro-inflammatory cytokines. We saw no evidence in this group here of classical inflammation or a major cellular infiltration other than this small increase in CD3 cells. And there was no tissue destruction. In other words, the tissue was not overtly or microscopically inflamed. And so the absence of overt inflammation seen in this study is consistent with the appearance of the gut mucosa in IBS patients. So our interim summary is that the microbiota from diarrhea-predominant IBS patients has the potential to alter gut function and behavior in a manner reminiscent of the donor phenotype. And these findings support a role for the microbiota in the expression of intestinal and psychiatric manifestations of this IBS subset. So our proposed involvement of the microbiota follows this model. There are risk factors: stress, infection, changes in diet and exposure to antimicrobials that changes the composition of the microbiota, that this then can affect the GI tract, as I've shown you, to alter gut function and produce GI symptoms, but it can also directly affect the brain through immune activation to alter behavior. So is the microbiota brain axis relevant to other disorders associated with altered brain function, particularly mood disorders and possibly degenerative conditions? So to examine this, we took microbiota from patients who have generalized anxiety disorder with no GI symptoms, took the microbiota and put it into a germ-free mouse. And we used a very simple test. The simple test involves putting 9 marbles on the bed of the cage. And normally mice leave these alone. But when mice become anxious, they start to bury them. And you can see here that the mice that were colonized with normal microbiota really made no changes. Whereas the mice colonized with anxiety, with microbiota from anxiety patients, buried at least 4 of the marbles. This is an easy way of assessing anxiety-like behavior. So we could transfer anxiety from patients who have a primary mood disorder characterized by general anxiety. And this has been replicated with anxiety and to some extent with depression in other studies. Similar work has been done involving Parkinson's disease and autism spectral disorders. The data, I wouldn't say that these are the strongest quality papers that I've seen, but nevertheless, they shed some light on the possibility that the microbiota may play some role in these degenerative neurological conditions. So what about therapeutic manipulation of the microbiota brain axis? Let's talk about probiotics. John Bienenstock and colleagues in Cork, Ireland, found that using a stress paradigm, that the Lactobacillus rhamnosus was able to attenuate the stress response by reducing corticosteroid levels. And it also, in a forced swim test, it attenuated the, as you can see here, it attenuated the stress response. This stress response, this therapeutic response of the probiotic was absolutely dependent on the integrity of the vagus nerve, because in vagotomized mice, you lost this effect. We did a study to look at anxiety. We used a model of a non-invasive nematode to produce anxiety-like behavior in a mouse. We used step-down tests, and we screened a number of probiotics. Here's the control. This is what happens in the infection. You get anxiety, this long latency to step down. When we gave Lactobacillus rhamnosus, there was a trend towards improvement, but it was not significant. However, when we gave Bifidobacterium longum, what we found was it normalized behavior, completely normalized it. And this effect was dependent on the integrity of the vagus. We then took that finding and moved it into man, and we conducted a pilot study, which was a placebo-controlled trial of Bifidobacterium longum, NCC 3001, on intestinal symptoms and behavioral symptoms in IBS 44 adult IBS patients with diarrhea or mixed stool pattern and mild to moderate scores in depression or anxiety. We gave 10 to the 9th CFUs on a daily basis for 6 weeks. And we measured symptoms, IBS-related symptoms and quality of life, as well as responses in terms of anxiety and depression. And we looked at brain activation patterns using functional MRI. As shown in blue, the responders to the probiotics was greater the improvement in IBS symptoms was greater, as shown in the blue bars, in those patients who received the probiotic compared to those who received placebo. However, when the probiotic was stopped, there was no difference really in follow. Sorry, this is the wrong slide. Here, we're dealing here with IBS-like symptoms and general abdominal well-being. The response rate was higher in the presence of the probiotic compared to placebo. But when the probiotic was stopped, the differences disappeared. So the improvement in abdominal comfort was totally dependent on active treatment with the probiotic. When we looked at depression, we found again that patients receiving the bifidobacterium had more patients responded in terms of significant reduction in depression. But in contrast to the IBS and abdominal symptomatology, this persisted for 4 weeks after the probiotic had discontinued. We next looked at functional MRI, and we simulated brain activity, the emotive centers, by showing them in a jittered manner presentations of faces portraying fear or smiling faces in a paradigm that's often used to elicit brain activity, particularly in the emotive areas. We can see that this produced activity in the red zone here, increased activity in the brain in placebo-treated individuals, but in the B. longum treated, the engagement of the brain, particularly in the amygdala, was subdued, it was a negative, and the degree of reduced engagement of particularly the amygdala correlated with the improvement in depression. So longum treatment for 6 weeks improved overall GI symptoms and depression scores. The effect on abdominal symptomatology was evident only during treatment. The antidepressant effect lasted for at least 4 weeks post cessation of treatment, and no effect of the probiotic was seen either on the microbiome profile or circulating cytokines or neurotransmitters. So what about fecal microbial transfer as a way of manipulating the microbiota-brain axis? There have been several studies in irritable bowel syndrome using FMT. Most of these showed mild beneficial effects in terms of IBS. There were clearly changes in terms of increased diversity in the microbiota, but only one of these examined changes in terms of behavior or mood, and one of them reported an improvement in depression. So this is an area that I think requires further work. So in summary then, these selected probiotic bacteria have the potential to alter brain function and may offer therapeutic benefit for the treatment of anxiety and depression. And based largely on preclinical studies, fecal microbiota transfer may influence brain function and offer therapeutic benefit for some psychiatric or possibly neurological diseases, but further work's required. And a cautionary note, because we can transfer components of behavior via the microbiota, all FMT donors, no matter what the indication, should be screened for psychiatric or neurological diseases in forthcoming studies. So to summarize the talk, the microbiota-brain axis influences brain function and behavior, not only during development, but in adulthood. Studies in a humanized mouse model demonstrate that diarrhea-predominant IBS microbiota has the ability to cause changes in both gut and brain function, reminiscent of the donor phenotype. thus justifying microbiota-directed therapy for this condition. A selected probiotic improved anxiety in the mouse model and was also effective in improving depression in a pilot study in IBS patients with comorbid behavioral changes. The ability of FMT to influence microbiota-brain axis does require further work. It's not ready for prime time. And microbiota-directed therapies may improve intestinal and behavioral manifestations of irritable bowel. So I will conclude by saying that the microbiota-brain axis represents a new and exciting frontier for the understanding and treatment of a spectrum of conditions that include IBS, psychiatric disorders, neurological conditions, such as Parkinson's disease and autism, among others. Thank you for your attention. And before I end, I would like to, okay.
Thank you very much, Steve, for this great talk. And I think that we can hold the questions for the Q&A session at the end for the sake of time. Before going to the next speaker, I would like to encourage the participants to place their questions through the Zoom chat. We have around 880 participants tonight, so a great number of participants. Please put your questions in the chat. So it's my great pleasure and honor to introduce the next speaker, Alessio Fasano. Alessio is professor of pediatrics at Harvard Medical School, and he is also chair of pediatrics at the Massachusetts General Hospital for Children. And we are very much looking forward to hear your talk. Alessio, please.
Thank you, Giovanni, for the kind invitation, and thank you for allowing me to join Steve in this very exciting seminar. You heard already some of the principles about the microbiome, that its involvement in the pathogenesis of diseases that affect gut-brain axis. And today we will discuss about celiac disease, and you will see that it's still pertinent to the gut-brain axis for some aspects, with celiac disease giving us some opportunity to study some more details in terms of environment-host interaction. Excuse me, this, oh, you go. Okay, so I would like to start, with the concept that is extremely important when it comes to chronic inflammatory disease and not an infectious basis. What you see in these slides, it probably is something you have seen so many times. That is what really start this hygiene hypothesis. Until the recent past, of course, the major reason why human beings get sick and develop disease and eventually die were infectious diseases. And with the advent of understanding microbiology and bacterial pathogenesis, we really went to war with these microorganisms, and we did a terrific job, particularly in the Western Hemisphere. And indeed, in only half a century, conditions that have been, again, plagued the human beings for 2 million years just plummeted thanks to the advent of hygiene information and implementation, the advent of antibiotics, of vaccines, and so on and so forth. So diseases like rheumatic fever, HPA, tuberculosis, mumps, measles just plummeted. However, and again, mainly in places where embracing the Western lifestyle, we saw a parallel and as steep as the decline was for the infectious disease, and so was the steep incline of a chronic inflammatory diseases like autoimmune disease, inflammatory bowel diseases, neurodegenerative disease, and metabolic disorders, and so on and so forth. Bottom line, we're not dying fast of infectious diseases anymore. We die very slowly of metabolic disorders. And this applies to many other conditions. And again, pertinent to celiac disease, where we were really convinced to have under control the environmental factor that is this protein grains as gluten. We saw, despite the genes were not changing, despite the environment, meaning the grains did not change, the same trend of this increase over time like doubling every 15 years of the incidence of celiac disease. These have been prospective cohort studies that show that this is a true increase of the incident disease.
I'm sorry, Professor Fasano, this is the webinar manager. A request from the translators. Can you pace your speech slightly?
I was trying to catch up a little bit the delay that we accumulated. But
no problem. I
understand. Sorry about that. But anyhow, a spontaneous example of an experiment of this, situation of changing lifestyle can change dramatically the incidence of this disease is happening in China. Not 50 years, but only in 10 years, China moved from a poor agriculture-oriented, economy to a Western, typical industrialization. And only in 10 years, they've seen an increase of, for example, food allergies that catch up only in that short period of time what happened in industrialized countries in centuries. So all this to say that lifestyle matters. And we have seen this also with cancer. Now that we know that there is a strong immunological component in cancer, and we've seen the same trend in this increase of prevalence in cancer in Western Hemisphere. But probably the most impactful and clear example of this quote-unquote epidemics of chronic inflammatory diseases has been seen with autism. In only a quarter century, we went from a prevalence of 1 in 5,000 to 1 in 58. And clearly, this demonstrates there is something, intrinsically wrong in what we have been doing that are fueling these chronic inflammatory diseases. And when I say we, I mean for people that embrace the Western lifestyle. See, if we want to see this phenomenon with a negative attitude, then we have to conclude that we are really changing the environment too fast for us to adapt. And this, detrimental changes leads to this increase of these diseases. However, the same phenomenon can be interpreted in a more positive way, because the old paradigm that suggested that if you are genetically predisposed to develop any of this disease, any of the chronic inflammatory disease, that's destined. You will eventually come down with that disease. It turns to be incorrect based on this epidemiological data, meaning that if we play our genetic cards wrong, as we've been doing in the Western Hemisphere last 50 years, then we will eventually favor the prevalence of these conditions. But if we learn a lesson what we've been doing wrong, then we eventually reshape our strategy and eventually play our genetic cards in a more wise way, try to mitigate, if not completely prevent, these chronic inflammatory diseases. So this implies that the old paradigm, genes plus environment, would be necessary, sufficient to develop disease, doesn't hold true anymore. There are at least another 3 elements that Steve touched upon already when he discussed the gut-brain axis in the context of IBS. One is the loss of barrier function, so permeability, particularly the gut, but also other interface with the environment. But the gut is the largest one, it's the more complex, so it's the one that seems to be more impactful. You lose barrier function and you allow these 2 elements, genes that live in our body and factors from the environment that in general keep at bay outside to actually physically interact. A 4th element, it's an immune system that is hyperbelligerent because, of course, these are all chronic inflammatory diseases, so there is a chronic inflammation as a common denominator. And of course, otherwise we would not be in this seminar here, the microbiome, that now we understand that it mechanistically can really be involved in all this, perfect storm, so to speak, to switch from genetic predisposition to clinical outcome. Even if 5% of these, 5 elements are artificially distinct, they are highly interconnected. So much so that we know that if you have an increased gut permeability, you will develop an imbalanced microbiome, what we call dysbiosis, and vice versa. An imbalanced microbiome can increase gut permeability. And increased gut permeability will give an uncontrolled access in our body of unknown self-antigen, and this will instigate the immune system to become hyperbelligerent. And in an immune system that is hyperbelligerent and create inflammation can increase gut permeability. So you got the concept of how everything seems to be interconnected. But ultimately, and I believe that is the most important concept to keep in mind, the impingement on the microbiome will lead to the epigenetic pressure on our genes. And that really dictates the state, the balance between health and disease by epigenetically changing genes that control, for example, our immune function or antigen trafficking that will eventually start our march from genetic predisposition to clinical outcome. Even look at the anatomy and the physiology of the gut will really be testimonial of the concept that I just mentioned. This is a cross-section of an intestine, happened to be in a mouse, but could be a human being. This big, black hole in the middle is the gut lumen, and there is a single layer of epithelial cells, this one in red, that is really what separates us from the environment. In the past, we thought that these epithelial cells were cement with each other, so that everything that pertains to interchange between us and the environment has to be negotiated through the cell. Now we know that in between cells, they are structured, they are highly dynamic, and can modulate their permeability that we call tight junctions, that we're going to revisit in a moment. But these epithelial cells, besides having all the machinery to digest and absorb foodstuff, they do much more than that. They have sensors on their surface that can understand if they are friends or enemies in the lumen, and if they're enemies, through these sensors that we call pattern recognition receptor, may eventually prime the immune cells to be ready to fight against an enemy. And depending on the immune cells, there are different, immune response that come out of it. For example, the one they are decorated in green, the dendritic cells, they are the first comers. They can even send periscopes into the lumen to understand who is the enemy that they have to face. And they can prime the other cells, like the one in blue here, the B cells, to produce the major weapon that we use at the mucosal level. They are immunoglobulins, particularly IgA, that can be secreted in the lumen and therefore fight the enemy outside the wall of our quote-unquote city, but also to decorate the intestinal microbiota, because the ones that decorated with secretory IgA will have a different function. So very important for that. Last but definitely not least, there are these white cells that's called the T cells, decorated in white. These are the heavy weapons, the one that generated the severe inflammation, either locally, if they stay there, or if they're programmed to leave the intestine anywhere else in the guts. This cartoon will show you the march from health to disease for what we understand in terms of mucosal immunology and biology. So start from left. This is the physiology. The junction in between cells are pertinent, so there is no increased gut permeability. There is a very tightly controlled antigen trafficking, and this induces mucosal tolerance, energy, or health. When we move to step number 2, in which, in general, because of dysbiosis, they are changing in the dynamic of these intercellular tight junctions that will become more open than they should be. This will increase antigen trafficking that is not now controlled anymore. And this will induce an immune response. And with that, inflammation. With the production of pro-inflammatory cytokines like TNF-alpha, interferon gamma, that per se can increase gut permeability. And now we are in this vicious circle of antigen trafficking, inflammation, more increased gut permeability, more antigen trafficking until you break tolerance and you develop disease. Which one? Well, depends on what kind of genetic background you have. You can develop, again, a functional problem, as you heard from Steve, if you're genetically predisposed to develop a visceral hypersensitivity like IBS. Th1 immune response, you develop a chronic inflammatory disease. Th2 response, you develop an allergic disease. Th17, autoimmunity, and so on and so forth. So the bottom line for what we've been discussed so far is that contrary to our previous belief, we are really made by two genomes. The human genome that now we know being relatively rudimental with only 25,000, structural genes. They are inherited from our mothers and fathers. They never change in their composition. So if we have genes that put us at risk for IBS, celiac disease, autism, Alzheimer, there is not much you can do. They're there. But they're not implied that if we have those genes, these genes will be expressed clinically. If they do or do not, depend on this other genome that is made by the microorganism. So the microbiome, if we're lucky, is inherited from our mothers and extremely dynamic, changes from one individual to another. In the same individual can change over time. If you allow me a parallel to express even more in depth this concept of epigenetic, imagine the genome as a piano with 23,000 notes, one for each gene. The piano is there, you can't change the piano. But there is no tune, meaning no disease will be played unless you have somebody that will sit down and play the piano. And that's the microbiome. It is extremely dynamic. So depending who sits the piano, let's say that to play the song celiac disease, you need to strike 300 notes. And now it's Elton John sitting at the piano that touch only 200, these 300 notes. So even if we are genetically predisposed, we are not playing the song celiac disease. But then something happened. We have a stress, we take antibiotics, we take a trip. In other words, the same condition that Steve was mentioning that started the march for IBS. This changed the composition and function of the microbiome. Now Chopin is sitting, he's a virtuoso, he touched all 300 notes, and then you have the clinical outcome. This again is emphasize the concept of how important is the composition and function of the microbiome in developing conditions that eventually will translate in chronic inflammatory diseases. I don't want to go too much in the details of how many microbes in the microbiota. There are a lot. There are at least as many microbes than the number of cells that we have, if not more. That there are many more genes that they generate. There are our own genes. They have an essential function for us, and that's the reason why we want to be in symbiotic relationship with, our microbiota, because they help to generate energy from food, regulate metabolism, produce essential vitamins that we do not have access to through the diet. They can regulate the immune system, as we mentioned before. They will fight against pathogens. Remember, penicillin is nothing else than a substance produced by some microorganism against some other microorganisms. However, in order to achieve all this function correctly and physiologically, we really need to establish a friendly, symbiotic relationship with our microbiome. And because we're all different from each other, even identical twins genetically can be different because of epigenetic pressure. It's intrinsic, the concept that the fit of the microbiome that will be beneficial for us, it's something extremely personal. This is the personalized medicine at its best. So there is no such a thing of the quote unquote normal microbiome. It's normal for that specific individual. It's the function that will not change from one individual to another. So we all need to maintain glucose level in our blood a certain range. Same for the blood pressure or creatinine levels. So that metabolic functions have to be common to all of us. How we achieve that depends on a perfect fit between who we are genetically and the microbiome that will live in symbiosis with us, so that we can obtain those physiological functions. If the microbiome goes off balance, these metabolic functions will be inappropriate, and this may lead to that march from genetic predisposition to clinical outcome. Here, it's just a snapshot of the many conditions that have been reported in literature in which microbiome imbalance has been linked to. But one key element that I believe that should also be stressed is that the engraftment, so the first encounter between the host and the microbiome and the maturation of microbiome, it's extremely important, I would say fundamental, for playing our genetic cards well. So mom's lifestyle, particularly in the last trimester, the way they were born, C-section versus vaginal delivery, the first 1,000 days of our lives from conception to the first 2 years of life will be instrumental to establish the normal physiological engraftment so we can find each other on a friendly ground as evolution planned, or if there are elements such as C-section, bottle feedings, use of antibiotics, in other words, variables that were not planned by evolution may change the course of this symbiotic finding each other that can have tremendous consequences later on. However, as also Steve mentioned, the vast majority of the studies on microbiome so far has been correlative. So you take a population of people that are healthy, a population of people with IBS, you find a different microbiome, and you make the conclusion that microbiome is the cause of the disease, in this case, IBS. That's not necessarily the case. There can be the other way around. Because you have IBS, for example, you change the motility of your gut, you can change your microbiome. So the microbiome changes the consequence and not the cause of disease. So there is a strong need to give credit to the entire field of the microbiome to move from correlation to causation. There are many possible approaches, and Steve again mentioned one of them by the humanized mouse models and so on and so forth. But when it comes to human studies, the best way to mechanistically link the microbiome with clinical outcome are longitudinal prospective studies, because that's the only way that we can move from association to causation. There are many advantages to do longitudinal studies because, first of all, you can collect samples before at the onset of disease, something that you cannot do with cross-sectional case control studies. You can enrich your cohort by choosing individuals that are at risk for a specific disease that you want to go after. There is a unique way to identify specific genetic risk when you use longitudinal studies, even if you have cohorts that are relatively small. And again, the multi-omic analysis that we would discuss in a moment is the only way that you can really do this in a mechanistic way is with longitudinal studies. And then, of course, if you choose wisely, and I will give you 2 examples, you will like to do birth cohort studies. So in other words, that you start from the engraftment until the onset of diseases. So, but you have to choose wisely. Why? Because if you choose Alzheimer, you have to follow people for 80 years, and that will be very, very difficult. And that's what we choose. 2 examples of 2 ongoing birth cohort studies, one very pertinent to gut-brain axis that is related to autism, for which we're not going to discuss. And the other one that is the object of my chat that, I was asked to cover today, that is celiac disease. This study is called CDGEM, that stands for Celiac Disease, Environment, Microbiome, and Metabolomic Studies. Why celiac disease is pertinent today topic? Because there's a lot that goes on in terms of gut-brain axis celiac disease. Many people that are affected by celiac disease, they have issues in terms of behavior or neuroinflammation that are testimonial of this gut-brain axis. And the other thing that is interesting as a model of celiac disease is that the old paradigm that I was telling you before, that has been always driven our approach to celiac disease, genes plus environment are necessary and sufficient to develop autoimmunity. Now, this seems to suggest that, and indeed, you go gluten-free, you subtract one of the two, and the autoimmune process is gone. Now, with the new events like this epidemiological data in which you see this doubling despite that these two elements did not change, makes us to believe that, yeah, they are necessary but not sufficient. Why? Because probably there are these other three elements that we were talking before that are at play: increased gut permeability, and a hyperbelligerent immune system, and an imbalanced microbiome. And that's what I want to focus on for the last part of my discussion. Focus on microbiome analysis in a longitudinal way to mechanistically link shift in composition and function with disease onset. What is known for microbiome of celiac disease so far? A lot of publication, more than 470 papers on microbiome of celiac disease, 300 which are involved the gut microbiome, 91 in children, but only a handful of prospective studies that are the ones that, in my opinion, are the only way to really mechanistically link microbiome with clinical outcome. There is another situation that needs to be said. A lot of case control studies have been done in which active celiac and people that are healthy controlled have been compared, and we have seen difference in the microbiome composition with some microorganisms to go up, some to go down. And in the first year of life, studies also been done to see what kind of microorganisms they change eventually when they develop later celiac disease. What are the caveats of these studies? Because there are several limitations. As already mentioned before, the cross-sectional design is very descriptive. One of the few sample standpoints, so it's a snapshot. There is no dynamic of the march from predisposition to clinical outcome by following the microbiome. Most of these studies are based on 16S sequencing. Again, 16S sequencing, it tells us only who is there. So in other words, what the microbiota is composed of, but not their function. And that's what we need to know. And we can do function analysis. The metabolic pathways that I told you are very important to dictate the phenotype and not surveil that way. And finally, several of these studies are done at different levels of the taxonomic level. There is a hierarchy of microorganisms, in this case, bacteria. And if you don't know at which level that particular study is, it's very difficult to compare one study and another. So one study is the phylum level, the other one is the order level, and therefore they are not comparable. But if you really want to reach the point in which Steve was talking about intervention by manipulating the microbiome, probiotics, fecal transfer, name it, you need to go to the species level because that's the target that you want to manipulate and correct when you talk about dysbiosis. And unless you don't do metagenomic analysis, so you answer the question, not only who is there, but what kind of function they are implementing in the gut, you're not going to reach that level. Not only that, but if you don't have robust clinical data, if you don't have genomic data, if you don't have epigenomic data, or metabolomic data, or the other omics, the virome, the parasitome, and so on and so forth, it is very difficult to put the work and put in context the science of microbiome for clinical applicability. So we really need to understand this complex interaction as the early steps of disease pathogenesis if we want to implement intervention, therapeutic intervention, or even more preventive intervention. So disease interception. That is our holy grail. So why celiac disease? Ideal. It materializes in the first few years of life, so there is not an open end, and is strongly connected to specific genetic markers, particularly the HLA. They're highly predictable if you develop celiac disease or not. In other words, if you don't have these genes, HLA, DQ2 or DQ8, the chance that you develop celiac disease is almost nil. So great luxury to stratify the population for these studies. And this can be done on a blood test, so non-invasive procedure. So in order to capitalize on this potential of celiac disease as a role model that can be extrapolated to many other conditions, we embarked in this prospective, multi-center, international observational study, this birth cohorts. As shown before, we enriched this because we choose to recruit 500 infants at risk of celiac disease, so somebody in family has celiac disease. And the goal was to identify validated specific microbiota metabolic profiles that can, predict on a specific genetic background who will develop celiac disease, and therefore to understand not only the role of the microbiome and the pathogenesis in the condition, but even the possibility to, again, implement primary prevention interventions. So this is the scheme of what was in our mind: 500 enrolls, 20% drop off, so 400 that will finish. Given our data, historical data, we predict 75% to be HLA compatible, so 280, and roughly 10% of them will develop celiac disease between 35 and 48. And using, the others that would not develop celiac disease as match control, we want to do this kind of studies. And the way that we did was with an intensive surveillance in the first year of life for a huge amount of metadata. So how these kids was born, again, pets in the household, other kids that were there, feeding, diary, exposed to antibiotics and so on and so forth. Very robust, and a robust, sample, collection over the years with a 5 years follow-up. What we have right now, we have recruited all 500. We have more than 80% retention rates. We collect something like 15,000 stool samples, more than 5,000 blood samples, 120,000 plus metadata, and also breast milk samples to look at the breast microbiome as a way to imprint, the child in terms of engraftment. So far, 31 of these kids developed celiac disease. You see the average age and the diagnosis. Well, let's start with the genome. We have whole genome sequencing of all the 31 kids that developed celiac disease and matched controls. And we were able to find the average of 4.8, 4.9 million mutants, variants, what we call SNPs, that we're seeing in kids that develop celiac disease compared to controls. Too many to work with. So we downscaled the number of mutants, variants to look at by reducing our analysis only at the genes that are expressed. So in other words, exons, and this brought down to roughly 185,000 mutants that we are now analyzing to see which pathways these genes are involved in, particularly immune system and cellular stress signaling and so on and so forth. And then annotating all these variants to see the biological function, to see if been already described in other autoimmune disease. In other words, so we have a clear understanding what it takes, genetically speaking, to really be at risk to develop this condition. And then, we add the transcriptome, metabolomic analysis, and of course, the microbiome. Why? Because predictive model for personalized medicine and primary prevention will be based on the microbiome function. Because if it's true that microbiome can epigenetically change our genes, this will translate in changing in genomics, epigenomics, transcriptomics, proteomics, metabolomics, ultimately the phenotype of the individual, and therefore the clinical outcome. That's the model that we have in mind. I'm going to finish up to just briefly mention the data that we generate with the first 10 patients and 10 controls that have been just published in PNAS and there's in press. This is a pilot study in which, again, we took 10 controls, 10 celiacs. We did the metagenomic and metabolomic analysis of these kids, and we analyzed more than 150 stool samples. We did both cross-sectional and longitudinal analysis, and we learned a great deal of lesson from these studies. So again, follow from birth all the way to the onset of celiac disease, and at the onset, we did the cross-sectional analysis, and then we went backward 3 months before the onset, 6 months, 9 months, and so on and so forth, to do a longitudinal analysis intra-subject to see what was the march of the microbiome composition and function in those that developed celiac disease and how that differed from those that did not. So bottom line, we know the genetics, we know the environmental factor when it comes into the picture because we have information when gluten is introduced. We know the onset of the disease, and therefore we can do this, cross-sectional and intra-subject analysis. How we did this? By analyzing the stool, metagenomic data, functional profiling, and then metabolic data on the stools. And we initially analyzed that independently. And, cross-sectional analysis identified some differences. Some microbes went down in cases versus controls. Some metabolites also changed. However, we could not find any meaningful diversity of microbial species of functional pathways that, can identify or segregate cases from controls. And the argument can be made, of course, you have only 10 cases and 10 controls. What would you expect? Well, when we did the longitudinal analysis, it was a total different story. First of all, we're talking about many more samples, because for each child, there were an average of 7 samples that were analyzed. And we were able to find in cases an increased abundance of microbes, pathways, and metabolites that have been previously linked to autoimmune and inflammatory diseases, while we saw a decrease, a loss of microbes, pathways, and metabolites that were previously reported as protective, have a probiotic-like activity. Plus, for the first time, we have seen previously unreported microbes, pathway metabolites that may serve for celiac disease-specific biomarkers of prediction of disease onset. While in the controls, we found increased abundance of protective, microbes and pathways and metabolites. That is pertinent to microbiome. However, we want to do a multi-omic analysis because our goal is to take all this information, genomic information, metadata information, microbiome information, metabolome information, and through machine learning, do personalized disease interception by creating mathematical modeling. This fellow is a synthetic biologist and computational biology expert, Ali Zomoroidi, that is leading this task, starting by taking a more comprehensive approach to microbes, functional pathway, metabolites analysis done in an integrated way, linked to host-derived omic data and clinical metadata, using machine learning to predict the onset of celiac disease. And only on 10 cases and then 10 controls, he was able eventually to predict in a relatively accurate way, 80% of chance, 9-12 months before the onset of disease, who would develop serious disease and who will not, just based on 22 species of microorganisms that were differentiating between cases and controls. So in other words, if this validated this with further machine learning redefinition of the data, it is possible with a certain level of accuracy to predict a year before the onset of the disease, based on this analysis of the microbiome, who will develop serious disease, who will not. Not only that, but there is also another element. Because metabolomic profiling is important as a biomarker for predicting who develops disease and does not, the underlying mechanism for the production of metabolites should take in consideration the interaction between one microorganism and another for the final production of metabolites. And also, of course, the diet that has a great deal of influence in that. Therefore, by recruiting this subject, doing the metagenomic analysis of these tools, and looking at the microbiome species that we isolated and their genome, we are able now to have a clear roadmap to understand which specific species and which specific strain can predict who develops serious disease, because we have sequenced them all. Once we isolate these strains, now we have their sequence, and we know an average of 140 microorganism bacteria per individual that will be able to use in an animal models to validate this data and so on and so forth. All this to create a personalized microbiota metabolic models. I'll give you just one example to finish, one case, one control in which we see metabolites produced by microbes in cases. They are in blue, and the ones that are produced in controls, they are in red, and we can predict who is doing what. When it comes to metabolites, they are linked to specific clinical outcome. What is next? We want to do deep metagenomic sequencing of stool samples for this additional 20 patients that we have to do more refined machine learning algorithm and training. We are doing single cell sequencing on immune cells. We will look at the alteration of intestinal permeability as the early step to break tolerance. Again, now we are looking also to serum metabolomic and proteomic profiling. We just finished, and we had another paper accepted for metagenomic and metabolomic analysis of the breast milk as the element for early engraftment and infant microbiome. We have isolated this strain of interest that are extremely valuable because can predict based on their presence who's going to develop serious disease and does not. We will use gut organoids to do this modeling beside animal models. And then, of course, we're going to focus on environmental factors that will predict, who's going to develop the problem. I want to finish again by thanking Giovanni for the invitation, the organizers, and all the entities that have been involved in supporting our studies. So of course, the CDGEM and their families to participate in the entire group of individuals involved in our studies. Thank you again.
Thank you very much, Alessio. That was fascinating and really inspiring. So I think that there will be questions later on during the seminar. Before we go to the Q&A section, I would like to remind the attendees that we have another seminar which will be held on Tuesday, June 15th at 8:30 p.m. Central Eastern Time. And again, we're going to have two great speakers there, Joël Doré from France. He will talk about the human microbiota, state of the art and the future perspective. And Yolanda Sanz from Spain. She will talk about dietary recommendations for promoting gut microbiota health. So we are very much looking forward to hear also this seminar. and the moderator of the webinar is Harry Sokol from France again. So now we're going to have a short promotional video from Bromatech, our sponsor, and then we will be back for the Q&A section of the webinar.
Bromatech welcomes all participants to this international event entitled Microbiota and Gut-Brain Connection, a new frontier in neurogastroenterology organized in partnership with the European Society of Neurogastroenterology and Motility. It is a pleasure and honor for Bromatech to sponsor this important and unique scientific forum. Bromatech is a biotech company that conducts research into the properties of bacteria since 1985. At the forefront of development for probiotics for human use, Bromatech is now an established company with a clear product offering for the natural treatment of the digestive system. The bacteria in the microbiome help digest our food, regulate our immune system, influence our behavior, and protect against other bacteria that cause disease. The strain specificity and efficacy of its probiotic products differentiate Bromatech from other companies. Mastering the natural attributes of nature for a harmonious life is Bromatech's inner motivation. Nature can teach us much more than we presently know. Microbes inhabit just about every part of the human body. We humans are mostly microbes. Over 100 trillion of them live in our body. Microbes outnumber our human cells by 10 to 1. The majority live in our gut, particularly in the large intestine. Over the years, Bromatech has built a research team that constantly analyzes the use and properties of bacteria that form part of the microbiome. Advances in genome sequencing technologies and metagenomic analysis, that is, the genetic study of genomes taken directly from environmental samples, have enabled scientists to study these microbes and their function, and to research microbiome-host interactions both in health and disease. Partnerships with universities and other research centers provide a valuable source of inputs, as knowledge transfer between academia and industry is an important driver of innovation. By means of laboratory testing and relentless gathering of clinical data, the company continually develops its own formulations. Scientific works and medical literature support our products. The human microbiome has extensive functions, such as development of immunity, defense against pathogens, host nutrition, including production of short-chain fatty acids important in host energy metabolism, synthesis of vitamins and fat storage, as well as an influence on human behavior, making it an essential organ of the body without which we would not function correctly. Bromatech has a long experience in microbiota research that allows it to understand the interrelation of different bacterial strains and dose the right quantity for optimal results. This is done through a well-developed methodology that combines laboratory research, clinical trials, and modern production solutions. Bromatech is committed to education. Over the years, it has regularly offered to the healthcare professional community multiple educational programs on the properties of bacteria and their interrelation with human health. To promote advancements in the understanding of the microbiome, it has founded congresses, seminars, academic courses, and scholarships. The microbiome is dynamic and changes with early development, environmental factors such as diet and the use of antibiotics, and in response to disease. The complexity and plasticity of the microbiota is important in maintaining homeostasis with the host's immune system and has an important impact on the digestive system. Speakers from different fields have had the opportunity to address participants sharing their clinical experience on the use of probiotics or presenting scientific research. An open debate is always welcomed at the end of each presentation, and participants stimulate discussions with their questions. Bromatech educational programs introduce medical professionals to the comprehension of this fascinating area of biology. Thank you for your attention and participation.
Welcome back. Before I said that the next webinar will be at 8:30 Central Eastern Time. It's Central European Time, so a little mistake there. So we can ask now our questions to these two speakers, and I would like to go through the chat because we received many questions from you. And the first one that I would like to ask to Steve Collins is whether there is an effect of stress on the microbiota. You alluded to that, but are there any strong data that suggest that stress can modify the gut microbiota?
So the answer is yes, but these are primarily from animal studies, because there you can control a lot of things that would influence the results. So yeah, I think the most robust studies are the ones performed by Michael Bailey with social stress, which was well controlled, showing a change in the composition of the microbiota. We published one using maternal separation that showed quite clearly that the stress involved in maternal separation can change the composition. We haven't studied function of the, or metabolic function of the microbiota in those circumstances. So yeah, but in humans, it's a very difficult experiment to perform. So most of the data where it's been studied have generated positive results, but these are, by and large, in animal studies.
Sorry, it was me. Alessio, you showed very nice new data about the predictive factors in the microbiota for the development of celiac disease. So we have a very specific question related to which strains are predictive of celiac disease. But before we go into that, I have a more general question. Do you think that these predictive factors allow us to intervene very early during the development of this disease? And can we modify these profiles in order to prevent celiac disease development?
I understand and appreciate the curiosity of the matter. Because this is impressed, unfortunately, I cannot give you the list of the strains, but it will be published in PNAS soon in a couple of weeks. So I just can't go to the list. However, and pertinent to your question, Giovanni, I don't think that the specific strain is important. I truly believe, the more that I work on this stuff, and it's been quite a while, the more I convince myself, and I would love to see Steve's opinion in here, the single strain or the composition of the microbiome, or what is the normal microbiome, is such a not appropriate question there. There is no such a thing. It's like to say, what is the normal length of your hair? It's so personalized. So it is the outcome of what is the interaction between the microbiome and the host, and specifically the metabolic pathways that are important to focus on. How you achieve that goal can be very different from one individual to another. So if we reach that goal, answer your question, the answer will be yes, provided that that analysis of microbiome is taken in the context, who is the host genomically speaking, and what metabolic functions they are changed that can predict who will eventually go on to develop celiac disease. That's the goal of this CDGEM, by the way, that if indeed we have this model that will be validated with additional machine learning, that, those 20 strains and those 3-4 metabolic functions are predictable of celiac disease later on, that's the target for the intervention.
Another question for Steve. I think that most of the data that you have shown during your presentation related to IBS with diarrhea. So the question is, first, do you think that the same connection between the microbiome and the brain occurs also for IBS with constipation, or is it something that is specific for IBS-D? And the second question is, do you think that a similar connection exists between the microbiome and anxiety and depression without changes in bowel function or without gastrointestinal symptoms?
Yeah, thank you. I can answer the second question first quite easily. We have data to show, and so have others published, that in patients who have, for example, mood disorders, that in some of those patients, you can transfer the mood, the alteration in mood to mice via the microbiota. So that's very in our hands when we study this in anxiety. Most of the patients we study seem to implicate a role for the microbiota in the expression of the psychiatric phenotype. When it comes to depression, our data would suggest that about 25% of patients who have major depressive illness but no GI symptoms have a microbiota that has the potential to transfer the behavioral phenotype. So I think it varies among these various conditions. Your first question was, is this axis important in other subtypes of IBS? We're in the process of studying that. All I can tell you is that, for example, we're studying at the moment patients who suffer from severe constipation following extensive antibiotic exposure or Clostridium difficile infection, or both. And we can transfer that slow transit into mice. But in contrast to what I showed you with IBS-D, we see overt damage to the, for example, interstitial cells of Cajal. We see significant macrophage infiltration of the gut. So I think that in these subsets, different mechanisms apply. We haven't studied behavior yet. We haven't found patients with severe constipation with enough psychiatric comorbidity to ask the same question in that subgroup.
Another question for Alessio. Alessio, you talked about the GEMMA study and the autism. Can you tell us a little bit more about this connection?
Yeah, I mean, again, there is a very flourishing literature on the microbiome and autism, of course. And, there is a dear need to really move, as I mentioned during my talk, particularly for autism, from descriptive to mechanistic studies. So we have this multidisciplinary team that was funded by the European Commission by this Horizon 2020 project, in which there's this public-private partnership. The idea, like for the celiac disease cohort, is to recruit 600 kids at risk for autism because they have a sibling with autism from birth, and start to really surveil these kids very early in terms of behavioral assessment. And as soon as we will eventually diagnose somebody with autism, we will have also an immediate intervention arm from one of our industry partner with use of prebiotics or probiotics. They are based on animal studies that were almost finished. They're very similar to the ones that Steve described for the IBS studies. So these are animals that have been humanized with the stools of children with autism with GI symptoms, children with autism without GI symptoms, and matched as a control to one of their siblings in the same household, meaning neurocompetent, with or without GI symptoms. So from these studies, we will have clear directions, mechanistically speaking, which component of the microbiome can really eventually be responsible, either because over, represented or, decreased, as happened for celiac disease. And therefore, the second part of this study is an interventional study in which, again, we will use prebiotics or symbiotics, so pre and probiotics together, as an intervention to really stop the progression of the neuroinflammatory process nowadays at the very early stage. And of course, the idea, like for celiac disease, is disease interception, so to prevent the next generation.
Thanks. Another question for you is related to the potential role of gut dysbiosis and gut microbiota with non-celiac gluten sensitivity. Any good data?
Very little data. There are some, again, descriptive data as we've been having with celiac disease, but we're very early stage there, so I don't think that we have robust data there.
Okay. Now we can ask another question to Steve that is related to fecal microbial transplantation. The data that were published in the literature were very, very heterogeneous. So in some instances, we got very nice and good clinical improvements. In other cases, we did not get any effect, or even a placebo. The placebo arm was even better than the fecal microbial transplantation. So where do we stand there? And is there any evidence that the donor is the key to understand the results that we get there? So do we have the super donors for IBS or not?
We haven't done studies. We haven't done a trial of FMT in IBS, and the studies, as you mentioned, that have been published are very, very different. Each study is different. It's different in terms of how the FMT is delivered, whether it's delivered colonoscopically, endoscopically, or in capsules. They differ in terms of whether the gut was prepared with antibiotics or with cleansing preparations. And it's very difficult to make, to draw any conclusions when you have such a heterogeneous group of studies. In terms of what we would advocate, I think that the passage of the transfer through the small intestine is probably going to be more important than administering, directly into the colon. In our studies in mice, where we've done things both ways, it seems that exposure of the small intestine, which is the house of the mucosal immune system in the gut, needs to see this transfer, this new microbiota, in order for it to have an effect. In our studies in IBD, we have certainly identified it's very much donor and recipient specific. We're conducting studies at the moment using machine learning to try to figure out what constitutes the best match. And we have indeed found a super donor where it seems that when that person donates microbiota, it seems to work very much better than with other individuals. So there are super donors out there, but generally speaking, I think it's going to be a combination of a match between the recipient and the donor. And we have got so much to learn in terms of what underlies that. I think before we can really start advocating this as a treatment.
I think that the background microbiota of the recipient is very important as well, because if you consider, what you are giving, it must be, taken up by the bowel and particularly by the ecosystem there, and probably even probiotics are different in terms of efficacy if they find different background microbiota. So
no, I agree entirely.
It's totally unknown, but we should know more about the permission of the resident microbiota to receive fecal microbial transplantation and probiotics and other type of interventions.
Yeah, I think we have to look very carefully at both the donor and the recipient.
Okay, we have many other questions. I think that we are getting to the end of the webinar, but maybe we can do an exercise here. I would like to end by asking Steve to ask a question to Alessio and Alessio to ask a question to Steve.
So Alessio, do you think that having a genetic predisposition is absolutely necessary for the expression of some sort of intolerance, doesn't have to be classic celiac disease, towards gluten? I mean, can environmental factors, particularly the microbiome, dictate and produce an intolerance without there being a genetic susceptibility?
I would say unlikely. Nothing is impossible in biology, Steve. You know that. But you know, you know, you need to have a genetic predisposition for your immune system to build up a Th2 immune response. Is a food allergy, or Th1 immune response is a chronic inflammatory process that needs to be put in motion by the microbiome. I truly believe that without the microbiome, even if you're genetically predisposed, that predisposition may be there for life and will never see the light of clinical outcome. And that's our goal. That will be our dream, eventually, to find a way to keep, you know, people in the tolerance, you know, mood in perpetuity.
Okay.
So my question for you, because you are a man of wisdom, knowing what you know for what you've been studying, but also for what you have not shared with us yet, and it's in your drawer, what would be, in your opinion, 5 years from now, the landscape of functional disorders in terms of gut-brain axis and the way that we would be able to manipulate them using the microbiota?
Okay. So I think there is a lot of mileage, even though we don't have the data yet, and we have a lot of reservations in terms of how to best approach this, that some kind of fecal, or let me say microbial transfer will do it. It's hard for me, even though we showed you data with one single probiotic improving depression. I have to say I was surprised. And in my own practice, I tend to move away from single microbiotic therapies for no matter what. I'm much more interested in preparations that have multiple bacteria, fermented products and things like that. I seem to get better. And I think that is the way that we will be able to, I don't think that identifying particular metabolites are going to constitute new treatments. I don't think that single probiotics are going to constitute new treatments. I think it's going to be some form of community transfer, or not single. But it's interesting because the probiotic that I showed you, which belongs to Nestle, is selling very well in terms of treating anxiety in dogs. So if you know that Purina is theirs, and I've heard many stories about people, particularly in the United States, who give their dogs this probiotic, and it turns them into a nicer animal. But that's all anecdotal.
That's right.
So this brings us to the end of this webinar, and we received many questions, but also many compliments to the speakers, and I would like to share this with you. It has been a great evening for us in Italy, morning for you in North America. Really, thank you very, very much for this webinar, and we are looking forward for the next one. Thanks, Steve. Thanks, Alessio. Have a nice day.
Thank you for having us.